There are three main steps involved in translation: initiation, elongation, and termination. So initiation will require many different components. You're going to obviously require the ribosome. Remember, the ribosome has a small subunit as well as the large subunit. It's going to require GTP. It's going to require your charged initiator tRNA. Remember, there's enzymes that are going to be adding the amino acid, the corresponding amino acid, to each individual tRNAs, and they need to be charged with that particular amino acid. In this case, since we're talking about initiation, there are going to be specific initiator tRNAs. Okay, it's going to be corresponding to methionine. We'll talk more about that a little bit later. Remember, methionine is always going to be the start codon for every single transcript. We're also going to need some different ions, things like magnesium, different initiation factors. We'll talk more in a minute about elongation factors. And of course, you'll need your mRNA containing the various codons. So to begin with, initiation in bacteria, this AUG start codon actually has a little bit of a sequence upstream, and we call this the Shine-Dalgarno sequence, and it's just an AGG. The reason for this is that it's going to base pair with the ribosomal RNA, specifically the small subunit of this. And this is going to help everything going to assemble around that mRNA, to assemble in the correct location as well. So then what's going to happen is the large subunit is going to combine along with the various initiation factors, the mRNA, everything's going to be localized at that start codon of AUG. So to begin with, just to visualize it, here you've got your mRNA binding to the small subunit along with the various initiation factors. Okay, here's the small subunit, here's this AUG, here is the initiation methionine. Next, this methionine will actually bind corresponding to this mRNA. Okay. The next thing that's going to happen, some of these initiation factors are going to be released. The large subunit is going to come along, and everything's going to assemble right around that start codon. We're going to talk a little bit, just a second, about this P site, the A site, what do those mean. But at this point, everything is going to be assembled, and you can now proceed with elongation. So in elongation, everything has to be already assembled. You need your initiator codon and your initiator tRNA and everything combined together. There are going to be several different sites. There is a P site or peptidyl site, an A site or aminoacyl site, and we're going to show you how those work in just a second. But basically, these charged TNAs are going to enter the A site. Another term for the A site, or a way to remember this, is acceptor site. This is where it's going to enter the complex. Peptidyl transferase is going to cause the peptide bond formation between the amino acid on the new tRNA, the one that just came in on that A site, and the growing peptide chain, which is bound to the P site. Again, P you can think for peptide, growing peptide chain. Once this reaction happens, there's nothing left on that tRNA that was in the P site. So that uncharged tRNA is going to move to the E site, the exit site. Everything shifts down. The tRNA bound is now going to shift over to that P site. A new amino acid can come in bound to a new tRNA. They're going to come in at the A site. This chain, this peptidyl transferase is going to transfer it to the growing chain. Everything's going to shift down, and this sequence of elongation and translocation is just repeated over and over and over. So to be able to see it, I think actually works a little bit better to see a video, so please make sure that you see the video that I added for this section. But to see it stationary, what you can see here is after initiation, you've got your initiator codon here. The second amino acid is going to come in on your second tRNA. And this is going to come in again at that A site. This peptidyl bond formation, or peptide bond formation, is going to be catalyzed. It's going to drop, you can see it's very small relatively here. It's going to attach it to this growing chain. So now we've got 1, 2. This reaction is catalyzed by peptidyl transferase. Now, this amino acid, that starting amino acid, is empty, and it is going to leave. It's going to be very temporarily bound in this E site or exit site. That first elongation step is now complete, and now you can continue it over and over and over. So now the second amino acid, or actually in this case the third, can come in. It can bind, and you can see here, this is what it would look like when it's bound. The peptidyl transferase is going to catalyze movement of these two amino acids to this growing chain to the new one here. After this happens, this one is empty. It is going to leave through the E site or exit site. Only 2 of these sites can be occupied at any one time. So once this one leaves, now the next one can come in, and you'll have many elongation steps, and so eventually your polypeptide chain can get quite long. Many proteins are going to be, I would say, the minimum that I would know of, maybe 50 kilodaltons. That's very, very small for a protein. Most proteins are going to be closer to maybe 300, 500, or even more. And what's going to happen then is it's eventually going to keep being read until it reaches a termination sequence. Remember the termination sequence, there's several different stop codons. We have a UAG, a UAA, or a UGA. Once any of those are found in the A site, this is going to signal this termination factor. We're going to have some GTP-dependent release factors. They're going to cleave that polypeptide chain from that tRNA, from the last tRNA, and everything is going to be released from the translation complex. So again, you can see here is a UGA. This is going to be a termination sequence. Everything is going to be removed, and everything is going to be disassembled. These components can then be read by, this mRNA can then be read by either the same ribosome or a new ribosome, and it can be used over and over and over. These ribosomes similarly can read the same mRNA, or they can read a different mRNA, just whatever they happen to come across next, along with the other initiation components. This empty tRNAs can go and get charged again with the new amino acids, and so on. Once this polypeptide leaves this leaves the ribosome, it's then going to be folded up into its secondary and potentially tertiary conformations. The structure of this is very important, and we'll talk about that a little bit later.